All right, let's get started. Um, for today, we have the subject of the generalized linear model. So it's another extension of linear regression. We will have 3 days in total about the generalized linear model. Today I'm going to only explain what it's for and how you can do it in R. We're not going to go into how you can interpret the estimates and all that stuff. That we're going to skip. We're going to postpone it until tomorrow. The reason being that you can all see that it's really just an extra addition to what you've already been doing, and there's just one more choice for the whole model selection procedure, so to speak. Okay, so the assignment of today, well, I don't expect anyone to be finished with all of the assignments of yesterday, so you can keep working on those. I also have some assignments at the bottom of this this chapter of the GRS course, 10.7.1, and you should have a look at that one. Even if you're not finished with yesterday's exercise, first have a look at this one, just so you know how to use GLM and so that you have some practice choosing the right probability distribution. Okay, then let's get started. This presentation isn't very long because, like I said, we're going to discuss most of it tomorrow, and then the day after tomorrow, Harold will be here because, in our experience, it helps to hear two different perspectives on the same subject for the more difficult subjects. Okay, so first I'll give a little motivation of why you would use a generalized linear model. So briefly recap ordinary linear models and the normality assumption. Then I'll explain, well, not really explain, I'll just show you what the exponential dispersion family is. And then I'll discuss some common types of data that you'll encounter very frequently in biological research. Then tomorrow we're going to talk about what a GLM actually is and what you can do with it. And I think we have 2 lectures that day, so I will also talk about how you can actually interpret the output and how you can get your output back on the original scale. Then on the in the fourth lecture given by Harold, he will talk about a special topic called over-dispersion, and there's also under-dispersion, but it's more rare. Okay, so ordinary linear models are just any model of the kind intercept plus slope times explanatory variable, and you can have multiple slopes in multiple dimensions, but this is the basic idea. And we fit these models by something called ordinary least squares, which means that you minimize the squared residuals. That's the estimating procedure, but that estimating procedure also comes with a PS error follows normal distribution. That little subscript is actually really important because this assumption of normality, only if that is approximately correct, are the estimates and the standard errors and the p-values you get from the model approximately correct. So what does it look like? These are the residuals of the same model. So it's just the vertical distances to the model. And we assume that if we account for all the explanatory effects, so if we estimate a linear relationship, then what is left behind, this error that remains, that follows a normal distribution. So in other words, if I flip it horizontally and I look at, some of them are close to the line, some of them are further to the line, some are positive and some are negative, and I can make a density plot of that centered around 0. And this is what we assume to be approximately normally distributed. Now, this assumption is really important because you have to have this assumption hold more or less in order to use the model for any kind of inference. So this is what I mean. If you produce a linear model summary like this, you really shouldn't be looking at any kind of p-values or the r squared or any interesting measures in here if this normality assumption is violated. This is also why we always do diagnostics. As it turns out, most things aren't normally distributed. There are things for which we can assume that given all the explanatory effects, that the remainder is approximately normally distributed. For example, adult male height in the Netherlands. If we know your country, if we know your sex, if we know you are within a certain age range, then there is still some variation in height, but we can assume most of it to be near the mean, and we can assume differences in either direction to be equally likely, so symmetry, and we can assume that further deviations are exceedingly rare, which is what the normal distribution says. But yeah, other examples like the number of birds in a park or the number of correct answers out of 20 questions, those things really aren't normal and also not approximately normal. For Poisson and binomial data, the two big differences are that, well, it's discrete, it can only take on certain values, whereas a normal distribution can take on any value in between, and they are bounded. The Poisson distribution counts cannot be negative, right? So this distribution stops at 0, which means that if the mean is close to 0, then the probability mass is pressed into the side, which causes this large right skew, right? There's a much bigger tail on this end than on this end. Same is true for the binomial distribution. If the chance of success for your number of trials is not very close to 50%, then there will be a skew, because again, it gets pressed into the side. And also for a low number of trials, even if it is very close to 50% the chance, then a normal distribution just doesn't approximate the shape very well, because there are only a certain number of possible outcomes. Now, then there are other types of data that still can be modeled with a generalized linear model, but that aren't part of this course. For example, the exponential distribution, which is a simple version of the gamma distribution. And yeah, that's basically any outcome that is positive, and it can have a right skew. So it can look like a normal distribution, but it cannot be negative. And it can also look completely like this, so it can be extremely skewed. Okay, so yeah, I think GLMs make more sense if you understand the history behind it, because there were a lot of people much earlier than the GLMs were used that were using Poisson regression, so regression specifically for count data, and logistic regression, where you're trying to estimate the chance that something will belong to either of two categories. So it's basically regression with a categorical outcome. And yeah, they tried to unify this. They tried to find some way to combine all of these methods into one overarching method. And that is what the generalized linear model is. And the way that they went about it is they found a algorithm they could use called iteratively reweighted least squares, an adaptation of the ordinary least squares algorithm. And that actually can fit the parameters of any probability distribution that we can write in this form. So this form is called the exponential dispersion family, and any probability distribution, if we can find, theta x and b of theta, a of, I should say phi by the way, but anyway, if anything you can write in this form, that you can also model with a generalized linear model with this method that they developed. So in other words, this is what the normal distribution actually looks like. This is the probability density function. And this is the one of the Poisson, which only has one parameter, lambda, which is both the mean and the variance. And a binomial distribution, so we have the number of trials, n, and we have the number of successes, k. All of them can be rewritten into this form. Now, how that works is not really important for this course. Just take my word for it that it can be done. If you want to see it, I can post it on Brightspace, but there will be no questions about it. But this is what you should remember. So the normal distribution is a logical choice for data that are continuous and that are symmetric around their mean. And remember that I said data, but that's actually not precise language. We make no assumptions about the data. We also make no assumptions about the response variable. We make assumptions about the errors. So I'll go all the way back to this earlier slide. The errors, the differences from the relationship that you estimate, that is the only thing that we make assumptions about. Now, if you can reasonably assume that to be approximately normal, then you can just use a linear model, everything we've learned so far. So continuous with a symmetric around its mean. Now, if you have count data, there are different kinds of ways you could model that, but for the purpose of this course, we model count data with the Poisson distribution, which is the distribution if your counts are truly independent of each other. Then finally, if you have a binary outcome, so that's logistic regression, where you have either of two classes and you're trying to estimate the probability of belonging to either class, and you can see how different explanatory variables affect that probability. Or ratios, like the number of people that survived out of the total number of patients, and hidden ratios, which is a type of data that is a ratio, but it's just worded in an unfriendly manner, such that it's a bit difficult to realize that it's a ratio. For example, what are the number of coronavirus cases, per population size? That's a ratio because there is a finite number in the population, so it's not really a count. Is this clear so far? Because this is really the only thing I have to say today. Okay. So in R, this can be done very simply. In base R, you don't have to load any packages. There is a function called GLM for generalized linear model. And the only thing you have to do to use this function is to specify the family of distributions that you find to be reasonable for your data. So for count data, you should just specify family is Poisson. There is more to this, but we're not going to do that until tomorrow. For today, it's just counts are this and ratios are this. Yeah? And of course, if your response variable is conditionally normal, you can just use ln. Okay, what does it look like? It looks almost the same as what we've been doing so far. So here I've entered some fake count data, and I have a grouping variable, and I have a treatment, which is control A or B. And then I fit a GLM on this, and this is a count, maybe it's the number of, mosquito bites or something. And you the only thing you do, you enter your counts as the response, you enter your explanatory variables, works the same way, just specify family and then choose the correct probability distribution, which is the assignment of today, choosing the right probability distribution. After that, it works the same as what you've been doing so far if the goal is hypothesis testing. So for today, we're not going to go any further than that. Just produce a summary and comment on which of these effects are significant, and you can write a conclusion about that. The estimates are a bit harder to interpret, but that's what we're going to do tomorrow. So this is what it looks like with interaction. Same thing as you learned yesterday, just add a multiplication sign instead of a plus sign, and then it will also estimate an interaction for your two variables. Okay, then for a ratio, it works a little bit different. You have to specify in your outcome, if it's a binary outcome, it works the same way, just response as a function of explanatory 1 plus explanatory 2, etc. If you have a ratio, you have to give R both of the numbers, and they have to be given in a certain way in two columns. The easiest way to do that is to use C-bind, which means column bind. So here I have the number of dead patients and a number of surviving patients, and just do C-bind dead, alive. And if you have successes out of n trials, then you use successes, and you do the number of trials minus successes, which would be failures. Yeah? It doesn't matter the order in which you do this, and that might sound really weird, but the reason is, you can either have the chance of having cancer or the chance of not having cancer, same model, it just flips the sign. Yeah, so this is the things that you should remember for today. What kind of probability distribution is most likely to be, appropriate for your data? How do you enter it in R? And then the summary, when we look at p-values, looks the same as yesterday. Okay, here's the binomial case with interaction. Just to show you, there's nothing different going on here if all I'm interested in is a hypothesis test. There are p-values here. You can see what the interaction looks like. You can see that group 1 is missing and that the control are missing. Why is that? In the output, I see a treatment A, treatment B. I don't see control. I see a group 2 and a group 3. Exactly. That's the group, that is the reference level. That is the one that's in the intercept. So here the intercept is group 1, treatment, control, and everything is compared to that. If that's not what you want to compare things to, not a problem, but then you have to use re-level, as we showed yesterday. Okay, So, yeah, that was kind of short, but that's on purpose because we have three things to do for today. I have a small quiz just to see if you understood what I said, or more remembered, I mean. Then, yeah, you have time to continue the exercise from yesterday, but first have a look at the exercise of today, which is choose the right probability distribution. I will quickly show it again. At chapter 10 called GLM. There are some videos here. They're from the course given in February, so you don't have to watch them, but of course you are free to. They're all the way at the end, 10.7.1. There are some exercises that are about here is some data, fit a GLM. You know how to do it. Just GLM and specify the right family, and then it will work. Okay. Last thing is the quiz. So here we have a couple of research questions, 8 I think, and the question is just what kind of model could I use for this? Could be a normal linear model, could be a type of GLM, just to see if it's clear so far. So let's take 5 minutes or so to read these, go over these, or 10, what shall we do? 2 minutes. Okay, let's take 2 minutes and then we'll go over them. Anyone watching online can answer their, yeah, they can give their answers in the comments if they want. Okay, shall we give it a try? Maybe another minute? How far is everyone? How many have you done? Like half, more than half? About half? Okay, let's just do 5 minutes and we can have a look at each. Okay, shall we give it a go? Okay. Okay, let's go with the first one. The effect of feed type on egg production of chickens. Anyone have an idea how you could study that? With what kind of model? Poisson. And why?
Because you count the egg.
Yeah, so a little bit of an assumption there, but of course there are different ways you could go about it, right? If you would measure the volume of the eggs, then it would be a continuous outcome. But if you measure the number of eggs that are produced with different feed types, then that would give you a count. You cannot count -1 egg or 1.5 egg. So in that case, you could use Poisson GLM. That's correct. Yeah, exactly, Basti. Okay, second one. Fluorescence is measured in GFP mutant cells at different concentrations of a reagent. What's the outcome in this case?
Fluorescence.
Yeah, so intensity or something like that. And what kind of outcome is that? Yeah, continuous. So we don't know if it's symmetric around its mean and also after accounting for the other things, but as a starting point we could use a normal linear regression. Turns out to be really skewed. You could use something like gamma regression, or you could use a transformation, but as a starting point a normal distribution would be fine. Okay, then the third one.
I'll say normal.
Sorry?
I would say normal.
Normal. A study assesses whether free-range chicken produce meat of higher protein content. And why do you think normal?
Because you would have a mean value and you would assess whether it produces on one side.
Yeah, so you have a certain amount of protein in your meat, and you know that could go up, could go down depending on the different, whether it's free range or the other class. And given the group mean, it's perfectly reasonable to start with the assumption that it's normally distributed. Yeah? We can't know if they're normally distributed, that's why we do diagnostics, but as a starting point that would be fine. Yeah, so someone in the comments says concentrations are ratios. Okay, you divide two things, but when I say ratios, I mean that they are bounded in two directions. So it must be the number out of a total. Yeah, and a concentration is not the number out of a total, unless it would be milliliters per milliliters or something. Okay, fourth one. Yes?
Binomial ratio.
And why?
Because you have per 100,000.
Yeah, exactly. So this is a hidden ratio, especially because a lot of news agencies, even papers, they would add a small print somewhere that says per 100,000. So yeah, when you see the number of cases, that sounds like a count, and it is a count. But when you say the number of cases per 100,000 inhabitants, then there is a maximum. You can't have less than 0 cases, and you can't have more than 100,000 cases. So this is a binomial outcome. Okay, how about the fifth one? A study measures the time elapsed before a bird enters its nest again after the nest is moved 5 centimeters. Yes? Sorry? And why? Okay, so that's this one is interesting. That would work if your outcome is seconds, because seconds are whole, right? But if you think of time as continuous, you know, you could measure in milliseconds, whatever. You could use a different probability distribution. So it's not wrong. It depends on how you design your study. But the thing is, you want to design your study such that it's maximally powerful. And the best way would, to measure time with as much resolution as you can. So go as far as you can. If you can have milliseconds, do milliseconds. And then we could, okay, there are limitations to the precision with which we can measure time, but we can assume the underlying phenomenon to be continuous. Yeah, and in that case, I would not go with a normal distribution in this case. it could work if you take the logarithm or something, but the problem with time is that maybe the first bird takes 1 second to go back to its nest, and second bird takes 2 seconds, 5 seconds, but another bird might be scared, might return after 5 minutes. And that this will always happen with these types of outcomes, where it's very strongly right skewed, because a bird cannot enter the nest after -1 second or something. So if the mean is close to 0 for the time elapsed, then it is going to be right skewed. And then, okay, this was a tricky one. You will not get something like this on the exam, but more likely candidate for this would be gamma distribution. Okay, next one. The effect of sex, age, alcohol consumption, and smoking behavior is estimated on cancer incidence rates. What is the outcome here?
Incidence rates.
Yeah, cancer incidence rates. And what kind of outcome is that?
500.
And why?
Because it's about the ratio.
Yeah, it's a ratio. So you'd have the number with and the number without cancer. So that would, starting point would be to use a binomial distribution. Okay, then the second to last one. The number of live bacterial colonies on a plate are counted after placing the plate in direct sunlight. How could we go about this? Yes?
Poisson.
And why?
Because you're counting bacterial colonies and -1 bacterial colony. So it's almost possible.
Yeah, so this one, he said Poisson, by the way. The wording is a little vague on purpose so that you can give your own interpretation. Let's say that there were no colonies on the plate before I did anything, and then I placed it in sunlight, and then I measured the number of colonies. Then we go from 0 to some positive number of colonies, right? So that will be a count. But let's say that this is a different type of experiment. I have a plate with colonies on it, and placing them in the sunlight will cause some of them to die. And I have some way, maybe coloring, to measure which ones are live, which ones are dead. And then again, we're at a ratio, right? So it depends how you set up the experiment. But if it's, if you start from 0, then it will be a count. If you start from a total number and you see how many are left, that would be a ratio of live to dead. Okay, then the last one. We have different species of flowers, and they're counted on 2 soil types to see which is more common where. And that follows what kind of distribution? What is the outcome here? I'll give you a hint. It's not written in the question.
The outcome be like frequency.
Exactly. It's the number of plants is the outcome. And that would be distributed as Poisson.
Poisson. Okay.
Counts are Poisson. Yeah, but if I say different species of flowers, like some sort of biodiversity study, you could say like, well, I'm interested in the different kinds of species. And sure, that could be. But just have species as an explanatory variable, then you can still do that. And then you have a nice count as an outcome, so you can use Poisson regression. Yeah, was this doable? Okay, then what I recommend you do is have a look at this first. So this is 10.7.1 of the book. You don't have to do all of them. There are some hard ones at the end. You definitely don't have to do those. But then, after that, try to do the exercise from yesterday that you haven't finished yet. Also the hard ones. The hard ones are not like what the exam is like, but try to see more data sets. Try to do this whole model selection thing a couple times. Because, well, first off, tomorrow I'll discuss the answers. And second, this is almost exactly what you're going to do for the graded assignment. You will get a research question, you will get a data set, and then you do an analysis, choose a model, do diagnostics, write a conclusion, just what we've been doing now. Yeah, so spend enough time on these exercises, and tomorrow I will discuss them. Any questions? Okay, good luck. Oh yeah, sorry, what time do we start tomorrow, someone asks? Let me check. It should be in the schedule, but I will look for you. Tomorrow we start in the morning at, wait, what's the day? Oh yeah, Tuesday. We start at 9:15 tomorrow. Yeah? Yeah, okay. Someone in the chat asked, can't a count be normally distributed if n is big enough? If you have very large numbers of counts, sure, it will be approximately normal. That is correct. But you will still have a model that predicts a continuous outcome. So Poisson regression is still a nicer model because it will predict a number. Yeah, so always go with the more sensible choice from a theoretical point of view. Okay, if anyone online still has questions, I'll keep an eye out on the Brightspace Q&A, and then I'll close the live stream.